Hello everyone, I am Joanna Moro, Bioactives Clinical Trials Specialist at Ingredia, and I'm going to introduce you to our new bioactive for blood sugar management, Pep2Dia. Pep2Dia is a unique patented bioactive that regulates blood sugar levels after meals. It is a natural hydrolysate of native whey with an active dipeptide AP, which inhibits the alpha-glucosidase activity. It is scientifically proven to lower blood sugar levels after meals, with no toxicity and side effects, and it helps maintain healthy blood sugar levels and prevents diabetes. We performed a clinical study in 2019 to test the efficacy of Pep2Dia. This clinical study was organized in two phases. The first one was an acute one, a crossover, double-blind, placebo-controlled randomized study with 7 days washout on 21 volunteers with glucose intolerance, with two groups, Pep2Dia 1.4 gram and a placebo group. And regarding the protocol, we gave to the volunteers Pep2Dia 15 minutes before breakfast rich in sugar, and we followed the blood sugar kinetics. Concerning the description of the population we recruited inside the clinical study, it was 21 volunteers with prediabetic condition. So with a mean glycated hemoglobin at 5.8% and a mean fasting plasma glucose at 1.1 gram per liter. The people were not under high pressure condition. They were overweight volunteers and above 14 years old. And we chose these criteria because we know that age and weight are among the main risk factors for becoming prediabetic. The primary outcomes measured is the area under the curve corrected by the mean fasting blood sugar, the IAUC. And we can see that after taking Pep2Dia 15 minutes before breakfast rich in sugar, we had a reduction of the area under the curve by 21%, which was significantly different from placebo. So Pep2Dia acted on the blood sugar after a meal rich in sugar among prediabetics. The second phase of this clinical study was an open trial, so 6 weeks of supplementation with Pep2Dia, with still 21 volunteers with glucose intolerance. And we asked no dietary and lifestyle habit changes in order to be sure that if we observe something, it will be linked to our products. The dosage was 1.4 gram before lunch, and we measured glycated hemoglobin after 6 weeks of supplementation. The glycated hemoglobin is the gold standard for glycemic measurements, and we can see that Pep2Dia significantly decreases the glycated hemoglobin after 6 weeks of supplementation, a key parameter to diagnose diabetes and glucose intolerance. So Pep2Dia decreases the risk to become type 2 diabetics. We performed another clinical study in 2020 to evaluate the dose effect of Pep2Dia. The protocol is the same, a crossover, double-blind, placebo-controlled and randomized study with 7 days washout. And we tested two dosages of Pep2Dia on 15 volunteers with glucose intolerance. Pep2Dia at 700 mg and Pep2Dia at 1,400 mg and the placebo group. And we can see that after taking Pep2Dia 15 minutes before breakfast rich in sugar, we had a reduction of the area under the curve with 700 mg and 1,400 mg, which were significantly different from placebo. So Pep2Dia is efficient at 700 mg on people with glucose intolerance. So thank you for watching this video, and if you have any question, please don't hesitate to contact us.